Hello, everyone. I'm Nicole Wilson, Program Manager for the United Mitochondrial Disease Foundation, and welcome to our Bench to Bedside virtual seminar series. This month, we will be hearing updates from various industry representatives and academic investigators on their mitochondrial disease clinical trials. MitoShare is UMDF's patient-populated registry, and the goal is to collect patient perspective with data complementary to clinical registries. Please follow the link below and share with your patient community. Give the gift of energy this holiday season. We're working to make Gavin's dream of a world where no one suffers because of mitochondrial disease a reality. To learn more about Gavin and his story, please click the QR code below. I'd like to welcome our moderators for today, Dr. Amel Kara with Massachusetts General Hospital and Dr. Bruce Cohen with Akron Children's Hospital. Thank you both so much for joining us.
Thank you, Nicole, and good morning or good afternoon for all of you joining us today. It's the December 11th UMDF Bench to Bedside series, and today we are going to hear from several industry representative and academic investigators about updates in clinical trial for mitochondrial disease. Our first presenter today is Dr. Natalie Yevgiohana, who is the co-founder and chief executive officer of Minovia Therapeutics. Natalie, welcome.
Thank you, Amel. It's good to be here. Thank you. We can move to the next slide. So Minovia has an open clinical trial that was approved in August recently by the Israeli Ministry of Health. Our clinical site is Sheba Medical Center, and the trial is for Pearson syndrome patients. It's a phase 1 open label, single dose clinical study to evaluate safety and efficacy of transplanting MNV201. Those are autologous hematopoietic stem cells enriched with allogeneic placental-derived mitochondria. The goal of the study is to include 5 patients, males or females, age 1 to 18, with a genetic diagnosis of Pearson syndrome and a body weight of more than 10 kilograms. The inclusion criteria will include patients with anemia or thrombocytopenia or leukopenia and or blood transfusion dependent, which means they receive blood transfusion at least every 6 weeks. Participants should be medically able to undergo the study interventions. And just next slides, please, just to remind everyone what that means. So Minovia's therapeutic approach is to use patients' own hematopoietic stem cells. So the process of undergoing apheresis, which is the collection of hematopoietic stem cells from the blood of the patient, should be approved by the medical physician. In that process, we collect the CD34 positive cells and enrich them with healthy and functional mitochondria from our cryobank. The process takes 24 hours, and then the cells are washed, packed into an infusion bag, and qualified to pass all the quality tests and be IV injected. This process is an autologous, fresh process, and this is why currently we can only conduct this trial in Israel, which is where our manufacturing facility is located. Next slide, please. So the primary endpoint for this study is safety, because this is a first in human with allogeneic mitochondria. And we will follow patients for 12 months, including all the blood tests and all the evaluations, and up to additional 9 years to include a questionnaire for safety of the patients. The secondary endpoint include, of course, the changes in anemia, thrombocytopenia, leukopenia, and blood transfusion dependency during the course of up to 12 months, as mentioned. We will also use the quality of life questionnaire, the IPMDS, like we have used in our previous trial. And we will look at frequency and less of hospitalizations during these 12 months post-treatment. Next slide, please. We have additional exploratory endpoint. This is the first time we can actually detect the transplanted mitochondria in the blood of the patients because we are using allogeneic mitochondria. And we developed the test to allow us to detect this allogeneic mitochondria. So that's one of the major goals of this study, to look at the pharmacokinetics of the mitochondrial DNA. We will also use some blood biomarkers to measure ATP production by PBMCs, respiratory chain activity, and mitochondrial DNA copy number, and of course changes in heteroplasmy due to the treatment. And we are also following anti-mitochondrial antibodies as a safety procedure to make sure that there is no immune response against the mitochondria. Our trial appears in the clinicaltrials.gov, and although it was launched in August, actually we enrolled 2 patients from Israel, but we could not treat them yet because of the war situation. And we really hope to resume the study soon. There is the contact information of the study coordinator in Sheba, Moran Levin. We collect information on patients, and hopefully we can start dosing as soon as possible. Thank you, everyone.
Thank you so much, Natalie. And as a reminder, due to the number of presenters today and the time constraint, we are not going to be fielding any questions. So we hope you understand. But if you do have questions, please forward them to the UMDF and we can get back to you. Our next presenter is Dr. Robert Fisher, the Chief Executive Officer and Chief Science Officer at Omas Therapeutics.
Thanks, Emma, for having me here at your webinar. Hello to everybody. Omas is a Berlin-based company here in Germany, developing a small molecule for mitochondrial disease. Next slide, please. Our molecule activates key regulators of cell metabolism and via these targets mitochondrial dysfunction and inflammation in patients with primary mitochondrial disease. We learned from previous 2 trials that our compound is very safe and very well tolerated in more than 160 individuals tested so far. Next slide, please. A specific finding from one of our previous trials was that our compound works very well, in particular in those patients which have elevated levels for GDF15. And GDF15 is interesting because it's one of the best biomarkers for mitochondrial dysfunction. And moreover, it's probably one of the best prognostic markers we have in several severe diseases. And recent publications showed that also in mitochondrial disease, GDF15 is linked to the severity of the disease. Next slide, please. That's why one of our main hypotheses of our first phase 2 study in primary mitochondrial disease is to test whether our small molecule OMT28, via its unique mechanism of action, is able to reduce GDF15 and other relevant markers of mitochondrial dysfunction and inflammatory stress in patients with mitochondrial disease. Next slide, please. So our study, which we are currently running, is a phase 2A study, and we enroll patients with myopathy and or cardiomyopathy. Patients should have mitochondrial tRNA point mutations and should all have, and also single mitochondrial DNA deletions are allowed. And on top, all patients need to have elevated GDF15 levels, which is a specific request for our study. Our aim is to enroll up to 32 patients in sites in the European Union. Currently, the trial is running in Germany and Italy, and we also plan to activate centers in the Netherlands. With regard to the design of our study, it's important to say that this trial is an open label study. So all patients will receive treatment in the duration of the study. It starts with an evaluation phase of 3 months where the patients are not yet treated, but we observe the developments of parameters without treatment. And then we have a second phase of this study with treatment of 6 months, followed up by a 4-week safety period. Primary endpoint is also safety and tolerability, as this is our first study in PMD patients, but we also look for the responder rate of patients reacting with a reduction of GDF-15 in the blood. And on top, we have a lot of secondary and exploratory endpoints, mainly biomarkers to learn about the effect of our compounds in mitochondrial disease. But also we have included all the relevant clinical endpoints for exercise tolerability, such as a 6-minute walk test, 12-minute walk test, 5-time sit-to-stand test, and questionnaires for quality of life, fatigue score, etc. Next slide, please. Yeah, here you see the centers which are currently all active and are recruiting patients. So far, we have 15 patients in the trial. So that means we are halfway of getting all the patients we need for this first study. Next slide, please. And of course, the results of this trial are expected in 25. We might see first results in the mid of next year. And based on that, we plan then to set up the further development and confirmatory studies as the second step. Thank you for your attendance.
Thank you very much, Robert. Our next speaker is Dr. Magnus Hansen. He's the Chief Medical Officer and VP Preclinical and Clinical Development at Aiva. Welcome, Magnus.
Thank you, Amel, and thanks so much to UMDF for arranging this meeting and inviting us. So at Aiva, our mission is to target the powerhouse of cells to improve the lives of mitochondrial disease patients, and we do this by developing new medicines for mitochondrial disease. Can go to next slide. So this is an illustration of our development programs, and I will focus on KL1333, which is a NOD+ modulator, and give a brief update on our phase 2 Falcon study, evaluating the efficacy of KL1333. So this study has been designed to be a pivotal study, which means that if it is successful, this study could enable an approval by the FDA and other regulatory authorities. If we go to next slide. So the Falcon study, it's a clinical trial in adults, and adults with genetically confirmed mitochondrial DNA-related disease. So it primarily focuses on two common and important aspects of mitochondrial disease, fatigue and myopathy. And we have two primary endpoints to evaluate these. One is a self-reported questionnaire for fatigue. So it evaluates fatigue symptoms and impacts on daily living. And this has been specifically developed for mitochondrial disease. And the other endpoint evaluates myopathy with the use of the 30-second sit-to-stand test. You move to the next slide. So the Falcon study is about a year long, and the study treatment is either KL1333 or visually identical placebo tablets taken twice daily. And 60% will randomly receive KL1333 and the other 40% placebo. And with the trial, we have tried to reduce the burden of participating as much as possible to enable a broad recruitment. There will be 5 mandatory clinical visits and then 3 additional visits that can be performed at home if wanted. The study also has monthly calls via phone to check in on the status of the participants. If we move to the next slide. So all patient-reported outcomes, such as the fatigue questionnaire, are reported using the participants' own phones or a separate device we provide if this is preferred. And this fatigue questionnaire is answered every week at the patient's own home, as we want to capture how the study medicine affects fatigue symptoms and impacts on daily life. And also other questionnaires are answered at home before traveling to each visit. And if you're interested to see the fatigue questions, the specific questions, they are available at this link via this QR code. If we move to the last slide, the Falcon study has now been recruiting patients at 18 sites in the US and Europe. And that's the first recruitment wave of the study. We closed screening of patients in October and expect to dose the last of this first set of patients before the end of the year. Results at 6 months from this first wave of patients will be included in an interim analysis around the middle of next year. And this will determine how the remaining study will continue and the final number of patients to be recruited. So if everything goes well and as expected, we will open up recruitment at these sites again, as well as open up additional sites after the interim analysis. So the specific inclusion criteria and contact info to the sites that are active can be found at clinicaltrials.gov or via our webpage. Thank you.
Thank you, Magnus. We are now going to hear from Dave Panicki, Chief Executive Officer of Tento Therapeutics. Welcome, Dave.
Thank you. Yeah, so very excited to have the chance to speak with you all today. If you move on to the next slide. Okay, so we're in a really exciting place right now. We've been working with the community and experts on studying our medicine in pyruvate dehydrogenase complex deficiency. The medicine is dichloroacetate. We have completed our clinical trial. We'll tell you a little bit about that. And we're preparing for our pre-approval filing meeting with the FDA. It's called a pre-NDA meeting, which we hope to have happen in Q1 of next year, followed quickly thereafter by filing for approval. Some of the other work that's been happening here is there have been a lot of questions about how many patients are there that really have pyruvate dehydrogenase complex deficiency. We conducted a pretty robust assessment and have come to what we think is the best number that we'll share today. We have partnered with Hope for PDCD, a patient organization that's done a diagnostic journey for us and talked to families with PDCD. We've supported emergency IND requests for typically newborn babies that have severe lactic acidosis and also pyruvate dehydrogenase complex deficiency. And we've just been honestly humbled by the families that participated in the study and everything that we've done, as well as there was a recent listening session with the FDA where they were trying to learn more about, what are the burdens that families and patients with this disease have. Next slide. So this is just a high level of our completed study. The first thing that we do is we screen patients to make sure that they have PDCD and meet our criteria. Then we genotype them. One of the things with dichloroacetate is it has a plasma level specific adverse event profile. And some patients metabolize the medicine more slowly than others. And so we screen them ahead of time to identify what dose is likely to be safe for them. And then we randomize the patients. It was a 9-month study. Patients were on placebo for half of it, and then they switched over to active for the other half. We targeted recruiting 30 patients, recruited 34. In that study, we measured functional outcome measures and biochemical outcome measures. And then we have an open label extension that's available to everybody. So of the 34 patients that entered the study, all 34 completed the 9-month trial and 32 continued on to the open label extension. What's exciting about that is this is a really devastating disease with severe outcomes and very early death typically in many of the patients. And so we're using the open label to actually characterize the safety profile and survival benefit that we think is associated with the medicine. So, the longest patient that we have in this is 1,100 days, and we're currently conducting an analysis versus some natural history studies to potentially show a survival benefit in our filing. So we have all of this for efficacy, and then obviously we've been monitoring safety, and the drug has shown us to be well tolerated. Next slide. As we mentioned, we looked at how many patients are there in the United States, and we conducted research with an external research organization that does this for lots of rare diseases. And essentially, if you look at PDCD, there's patients present either, in infancy, typically within the first weeks of life. There's another group of patients who are a little bit less severe. They overlap a lot with, have a co-diagnosis with Leigh syndrome typically. These are patients that present sort of sometime between 1 and 12 years, typically on the 2 to 4 year range. And then there are older patients that present later in life with symptoms that are diagnosed. What we see here is about 1,000 of the 2,000 believed patients in the United States are diagnosed. The diagnosis rate is much higher for infantile than for childhood disorders. And the childhood disorder, or the childhood onset of the disease, is typically associated with misdiagnoses like cerebral palsy and other potential indications in which patients are, treated with a disease, treated for their symptoms, but don't have the underlying disease. So should we be approved, that's a place where we're going to hopefully go out and help educate physicians to, do more genetic screening diagnoses and hopefully help make those available for a cheaper cost as well. Next slide. And as I mentioned, the FDA PDCD listening session was something that we thought was really important. With all of these rare diseases, it's really difficult for the agency, who's got to take in, all of the challenges and potential, phase 3 or pivotal data about, what are the needs of the patient communities? How are they able to deal with it? How severe and debilitating are these diseases? And so they took the time to do that. Industry is not a part of these. This is just patient communities. Dr. Ganetsky from CHOP was attending as well. And the key messages that, I think these brave families shared was PDCD is devastating and progressive. The current treatments, which are primarily the ketogenic diet and thiamine, are not enough. Families are doing everything that they can here, but they really need approved treatments, and they need, the ability to sort of move science forward here. So from our perspective, this was really helpful because we do plan on, filing our NDA in the coming year, and, think that this is an important piece of education and context that the agency has the opportunity to see. And with that, I believe that's the general update that we have. So, full speed ahead for the SAIL team. And thank you for your time.
Thank you so much, Dave. We are now going to hear from Dr. Jan Smitnik, Chief Executive Officer of Kreon. Jan?
Yeah, thank you very much, Amel. Nice meeting you all. As you know, Kreon is a Netherlands-based biopharma company. And it's a pleasure for me to update you on both our clinical status and the plans for the future regarding our lead compound, sonnecombinol, and our initial target indication, patients suffering from the M324A2G mutation, including classical MELAS, maternally inherited diabetes mellitus and deafness, and mixed phenotypes. May I have the next slide, please? So this is a cartoon of our lead compound called sonnecombinol, also known as KH176, and the compound targets the key underlying mechanisms in primary mitochondrial diseases. It's a reductive distress modulator, so it helps to restore the cell's metabolism. It's an oxidative distress modulator, and it prevents ferroptotic cell death. And next to that, it's also an inhibitor of what is called the microsomal prostaglandin E synthase 1, which result in anti-inflammatory effects. May I have the next slide, please? So, as mentioned in the introduction, our initial target mitochondrial indications are patients suffering from the M324A2G spectrum disorders, of which approximately 5 to 10% suffer from classical MELAS, and the remaining from either maternally inherited diabetes mellitus and deafness, CPO, and mixed phenotypes. As you're all aware, these are progressive diseases resulting in serious multi-system symptoms, in particular involving organs and tissues with high energy demands, like the brain and the central nervous system. Next slide, please. So this is an overview of the clinical studies so far performed and completed. So we started off with a phase 1 classical SAD and MAD study. Then we executed a proof of concept trial, a phase 2A trial in 18 patients. And that was followed by an integrated phase 2B program. And that integrated phase 2B program consisted of an randomized controlled study involving 27 adults with the 3243 mutation, and patients were offered to continue treatment with sonicomanol in an extension phase, which in 2 countries had a duration of 52 weeks, and in another 2 countries now of 78 weeks. The next slide, please. Yeah, this is a high-level summary of the phase 2B program, so consisting both of the RCT and the extension studies. Sonicomanol showed good tolerability and a favorable safety profile up to 78 weeks of sonicomanol treatment. An additional group of patients already entered into a named patient program, so we now have up to 2 years a good tolerability and a favorable safety profile with sonicomanol. We showed in the phase 2B program statistically significant and/or clinically meaningful results in multiple relevant outcome measures. And those outcome measures included fatigue, muscle strength, pain, and quality of life. And in line with the expectation, longer-term data, of course, showed more pronounced effects than shorter treatment periods. So supported by both the favorable benefit-risk profile, sonicomanol is now progressing towards a phase 3 registration trial for patients with the 3243 mutation, including classical MELAS, MRDD, and mixed phenotype, and a selection of best performing outcome measures of the 2B program have now been prioritized as primary and secondary endpoints of a global phase 3 study. And we're currently finalizing the preparations and initiation of a registration phase 3 study. Have the next slide, please. And with that, I would like to thank the UMDF and wish everyone a wonderful holiday season and already a happy new year. Thank you very much.
Thank you, Jan. And our next speaker is Dr. Fernando Scaglia. He's the professor at the Department of Molecular and Human Genetics at Baylor College of Medicine and Texas Children's Hospital. And he's going to tell us about his investigator-initiated clinical trial.
Thank you, Mel. And I also want to be very thankful to UMDF for this opportunity to present this. This is a review on the phase 1 dose finding and safety study of L-citrulline treatment for nitric oxide deficiency in MELAS. You can advance to the next slide, please. Yeah, so as we know, this canonical mitochondrial syndrome has the most common pathogenic variant in M3243A2G in MTTL1, and it has been hypothesized that altered metabolism of nitric oxide may lead to changes in the lining of blood vessels, decreased perfusion, and stroke-like episodes. And IV arginine supplementation has led to clinical improvement if given within 3 hours of onset of symptoms. Moreover, patients with MELAS have been found to have hypocitrullinemia. Next slide, please. So based on previous studies, stable isotope studies that we conducted at Texas Children's Hospital on adults and children with MELAS, we have hypothesized that they would have impaired nitric oxide production rate, and that arginine and citrulline, since they are nitric oxide precursors, would increase nitric oxide production. And citrulline supplementation would increase nitric oxide production to a higher degree than arginine supplementation. And this is for like several factors. And currently I don't have the the time to like go over them. Next slide, please. So we initiated, this was actually initially a pilot project from NANDEC, and turned out into a phase 1 clinical trial to evaluate the primary endpoint is to look for the maximum tolerated dose of L-citrulline in Milas. We obtained an IND from FDA. We obtained NIH approval. This has the following NCT number, and these have been the inclusion criteria. So clinical diagnosis of Milas with neurological or muscular symptoms, including metabolic strokes, the age of participation, 18 through 65 years, and all participants with the current pathogenic variant, 3243A2 gene, MTTL1, elevated plasma lactate taken at any point in the screening period, which goes from 6 months prior to the screening visit, including and up to the baseline visit, and a score of 26 or higher on the Montreal Cognitive Assessment, and cessation of arginine, citrulline, or compounds affecting nitric oxide 1 week prior to the study. Next slide, please. So the primary aim, the primary endpoint has been to establish as a safety study the maximum tolerated dose of L-citrulline in initially 24 adults with Milas at one of the following doses: 10 grams per day, 20 grams per day, 30 grams, or 40 grams per day, divided 4 times a day for up to 1 month, with an additional month follow-up safety visits at week 6 and week 8. And then we have been looking for primary safety outcomes or those limiting toxicities, which could be associated to the overproduction of nitric oxide, such as hypotension, gastrointestinal symptoms, and migraine headaches. Next slide, please. So the secondary aims of the study have been by using like ASL, MRI, to determine the cerebral blood flow and cerebral blood vessel reactivity, and to measure plasma amino acids at baseline, week 1 and week 4, to determine citrulline and arginine levels, and plasma lactate and plasma alanine at baseline, week 1 and week 4, as disease biomarkers, and measure plasma guanidino compounds via targeted metabolomics at baseline and week 1 to determine potential arginine toxicity. Next slide, please. And this is the study design. We have collaborated with a statistical team in Columbia University in New York, using the time to event continual reassessment method to establish the maximum tolerated dose of L-citrulline. And the participants receive the dosage, and this is determined by this continual reassessment algorithm. And the study participants are enrolled one at a time serially. And the first subject receives L-citrulline dosage of 10 grams a day for up to 1 month or until a safety outcome is reached. Next slide, please. So the dosage for each subsequent subject enrolled is either increased or decreased based on the determination of this algorithm, which takes into account the rate of safety outcomes encountered by previously enrolled subjects to determine dosage for subsequent participants. After a participant receives study medication for 1 month or encounters a safety outcome, the administration of medication ceases. Next slide, please. And this is like a summary of the assessments. There have been like weekly visits, including after the screening visit, baseline week 1, week 2, week 3, and week 4. So the participants have been on citrulline for 1 month, and then follow-up safety visits, week 6 and week 8. And this is actually the list of all the laboratories that are collected and the diaries that are collected from the study participants. And there have been weekly monitoring phone calls for each participant to assess their safety. Next slide, please. So the trial has proceeded under an NIH and IRB approved protocol, full engagement from the clinical and statistical teams, our independent medical monitor, the data monitoring coordinating center, and under the guidance of the DSMB. And we have conducted weekly meetings during this 2.5 years that the trial actually has lasted. The last data safety monitoring board meeting was held on March 9th, allowed to continue. We submitted an annual review to the FDA on April 4th. 10 participants were enrolled in the study with no screen failures, and all of them have completed the study. And it has been determined that we have enough data with 10 participants to really conclude the study. So now we are proceeding with the CRA analysis by the clinical research associate, and one is that is done. We will proceed with data log and data analysis. And eventually, of course, we will be presenting this at other webinars at the UMDF meeting and other national meetings and proceed with manuscript preparation. Thank you so much. You can proceed to the next slide. Thank you.
Thank you, Fernando. Up next is Dr. Magali Tael, Chief Medical Officer at Gite Biologics.
Thank you so much for the invitation. And next slide, please. This is our disclaimer. Next slide, please. So we are developing gene therapies in inherited retinal diseases. Next slide. And I would like to focus this presentation on Lumivoq gene therapy development in Leber hereditary optic neuropathy. And next slide, please. As you may know, LHON is a maternally inherited mitochondrial disease. It is a rare disease that affects the retinal ganglion cells. Patients have a dramatic drop of vision, firstly in one eye, and a few weeks later in the second eye. And within 1 year, the patients become blind. Different mutations on the mitochondrial DNA can lead to LHON. The mutation on the ND4 mitochondrial gene is the most severe mutation. Next slide, please. The gene therapy Lumivoq specifically targets the causal mutation, the mutation on the ND4 mitochondrial gene. And Lumivoq gene therapy is a recombinant adenovirus-associated vector type 2 containing the healthy mitochondrial ND4 DNA. Lumivoq is injected via intravitreal injection, and we have an unprecedented data set with 252 ND4 patients treated with Lumivoq. Next slide, please. So 3 phase 3 trials were completed, and 1 phase 3 trial is ongoing. Next slide, please. So in the phase 3 studies, 174 patients were treated with Lumivoq. Approximately 70% of patients had a remarkable improvement of visual acuity of at least 3 lines from that year. And importantly, patients unilaterally treated with Lumivoq showed a bilateral improvement of visual acuity, meaning that treated and untreated eyes similarly improved. It explains why the primary endpoints were not met, as the primary endpoints were the comparison between treated and untreated eyes. So this improvement of visual acuity for untreated eyes is related to the contralateral therapeutic effect of the drug. Next slide. And this contralateral therapeutic effect on untreated eyes has been explored. Next slide, please. So recently, in 2 ND4 patients unilaterally treated with Lumivoq, we demonstrated the transfer of Lumivoq DNA from the injected eyes to the contralateral uninjected eyes. And this transfer of the vector DNA between eyes appears to be the main mechanism explaining the contralateral effect observed in patients. So as the contralateral effect eliminated the control group formed by the untreated eyes, the untreated eyes are not a control group anymore and can be considered as treated eyes. Next slide, please. Therefore, to assess the efficacy of Lumivoq, the only solution is to compare Lumivoq data with an external control group. Next slide, please. So we performed a predefined statistical analysis to compare Lumivoq data to an external control group using exclusively individual ND4 patients' data. In the treated group, 174 ND4 patients from the phase 3 trials, and in the external untreated control group, a total of 11 natural history studies were identified, leading to a group of 208 untreated ND4 patients. Next slide, please. So this graph represents the course of visual acuity over time using lowest curves modelization. For the 208 untreated patients in blue, we observe the absence of visual acuity recovery over time. For the 174 Lumivoq patients in pink, the dynamic is totally different. Over the same period of time, we observe a progressive and sustained improvement of visual acuity. The difference in visual acuity enlarges over time and is clinically meaningful. Next slide, please. We are now moving to safety. Next slide, please. We have a lot of follow-up data with the project. In total, 252 patients were treated with Lumivoq. Lumivoq is well tolerated. There are no systemic safety issues as the biodissemination of the product in the blood is negligible. The main ocular adverse event is intraocular inflammation, which is classical with the gene therapy. The good thing with Lumivoq is that this intraocular inflammation is mostly mild, is responsive to conventional treatment, mostly corticosteroid eye drops only, and is resolutive without sequelae. And importantly, the good safety profile is comparable in unilaterally and bilaterally treated patients. Thank you for your attention.
Thank you, Magali. Up next is Dr. Alana Sullivan, the Senior Clinical Development Scientist at Stealth Biotherapeutics.
Thank you, Amel. As Amel just said, I'm Alana Sullivan, the lead clinical scientist for the New Power trial, and I'm very happy to be here to provide you with an update today. But first, I'm going to start with a little bit of background on Stealth Biotherapeutics and the program in which the New Power trial resides. Next slide, please. Stealth Biotherapeutics has 15 years of focused experience in the clinical development of mitochondrially targeted compounds. Our mitochondrial platform is diverse and spans across several indications and assets. Today, I'll focus on our lead investigational compound, elimipritide, which has been studied in over 1,400 patients and healthy volunteers, with long-term treatment duration being up to about 5 years. Next slide, please. So clinical experience with elimipritide and PMM is not new to Stealth, as Stealth has completed several early phase trials, and of course, most recently, the EMPOWER-3 phase 3 trial. From learnings across different programs, Stealth has incorporated several ideas into the study design of our current New Power trial, which is a phase 3 pivotal trial. Next slide, please. This is a schematic of the phase 3 New Power trial, MD301 trial. Subjects having PMM caused by pathogenic nuclear DNA mutations, and specifically replosome-related mutations, were enrolled. They then were randomized in a 1-to-1 fashion into either an elimipritide treatment group, where elimipritide at a 60 milligram daily subcutaneous dose was administered, or placebo in the same manner, for up to 48 weeks. We kept our primary endpoint as the 6-minute walk test and brought back 2 functional endpoints as stated here, the 5 times sit to stand and the triple time up and go. We also included a new secondary endpoint of the PGI-S scale, which is a patient global impression of severity scale. The trial was fully enrolled in October 2023. Next slide, please. And here are some key study metrics associated with that. Our first patient in was in April of 2022, and we have a projected last patient out in the fall of 2024. Base lock around the end of 2024, and completion of study around the beginning of 2025. Global enrollment was reached at 104% of the primary population target. This spanned across 10 countries with 34 sites activated. 94 of the patients randomized had replosome-related mutations, and 8 had non-replosome in DNA mutations. Thus far, 14 subjects have withdrawn from the study, and actually, as of today, this has upticked from 18 to 19 subjects being completed treatment per protocol. Next slide, please. We anticipate our top-line data to come out around the end of 2024, And while we don't have time for questions here today, I welcome questions being fielded through UMDF. Thank you very much.
Thank you so much, Ilana. Our next speaker is Dr. Susan Van Meter, Program Physician at Rare Disease Organization at UCB.
Thank you for inviting UCB to present today. Next slide, please. So this is just a standard disclosure slide that our legal team requires us to present. But basically, I'm an employee of UCB. The drug is an investigational product, and it has not been approved for any indication by any regulatory agency. Next slide. So when we take a step back and try to say, how do we evaluate a treatment for an ultra-rare disease? The goal is the same, whether the disease is rare or not. You have to show clinical benefit and safe use. The difference comes in terms of execution and the challenges to that in terms of, what is the disease natural history? You've heard today several companies have set up their own natural history studies to get better information on this. The diseases are often very heterogeneous in their presentation. Sometimes there's a very undefined or poorly defined regulatory approval pathway. There's relatively small numbers of patients that are available to participate in the clinical studies, and these patients are often quite geographically dispersed. Next slide, please. So UCB is investigating a treatment for thymidine kinase 2 or TK2D deficiency. And ultimately what happens is that there is a decrease in energy for cells, and ultimately the cells that require a lot of energy, such as the muscles, tend to be the most impacted. I won't necessarily, in the interest of time, go over everything, but we do know the specific pathophysiology for the disease. It involves the thymidine kinase 2 enzyme, and understanding this has helped in our development program. Next slide, please. So the drug that we are investigating is called doxycytine and doxribtimine. Some of you may have known it as MT1621. It is a one-to-one combination of two drug substances, deoxycytidine and deoxythymidine. Next slide, please. Sorry, it looks like this is a bit of a build. You might need to press advance again. The basic mechanism, okay, well, if you could go back to the previous slide. Looks like there's a little bit of some elements missing, but essentially the middle part represents the mitochondria. The way that the drug works is by utilizing any residual TK2 enzyme activity, and outside of the mitochondria, using alternate pathways to help facilitate mitochondrial DNA production and ultimately energy production for the cell. Next slide, please. So the very first investigation of treatment with deoxycytidine and deoxythymidine was the MT16-101 study, or the 101 study. And this involved a review of 38 patients. It was a retrospective study. It involved a chart review of 38 patients treated with a drug that were alive as of the last date of contact. Well, actually, it enrolled patients and evaluated their symptoms from onset of disease symptoms through the last day of contact. As of the last day of contact, all treated participants were alive. in order to evaluate survival context, an untreated patient pool was developed by examining the literature. So reports of patients treated in the literature were carefully reviewed, and a unique cohort of patients were set up for evaluation of survival. Very nice models showing improvement, very robust survival benefit of treated patients versus untreated patients. Unfortunately, the small sample size and the absence of any deaths in the treated participants result in a very large confidence interval. So this certainly suggests a treatment benefit, but it lacks the power to really definitively demonstrate that. So the company has undergone further work. Next slide. This is just a summary of adverse events. The most common treatment emergent adverse event was diarrhea. Most of the adverse events were mild or moderate in intensity. We had 2 patients discontinued due to elevation of transaminases. Next slide, please. So as I alluded to, we've been able to pull together a lot of different sources of information to put together a registration package. First, because we know the pathophysiology of the disease, we were able to use non-clinical models to confirm target engagement with increased mitochondrial DNA copy numbers across most tested tissues, and specifically skeletal muscle, and the ensuing mitochondrial biochemical changes with increases in respiratory chain complexes. We've used all available data. So we rely very heavily on real-world evidence. Our untreated patients are not coming from a randomized controlled trial. We have an untreated patient group that has been set up using primarily patients from the literature and some patients that we evaluated in a subsequent chart review study, the MT1621-107 study. Treated patients build on the initial findings in the 101 study to evaluate survival and change in disease trajectory in non-survival endpoints. We've used every potential regulatory interaction that was available to us. For the FDA, we have breakthrough therapy designation, and in the EU, we have PRIME. These have allowed greater contact and discussions with regulatory agencies to work on a pathway to demonstrate the substantial benefit. Next slide, please. So where are we now? We are continuing with the development program. The targeted population is patients with an age of symptom onset on or before 12 years. We have a compassionate use program. We receive requests from treating physicians. These are evaluated on an individual basis. We continue to support the mitochondrial disease community. We're in the process of collecting additional data in the patients enrolled in the clinical development program to address FDA's pre-submission questions. And we are planning submissions in 2024 to both the European Medicines Agency and FDA. Next slide. Without the patients, families, and physicians, we would not have a development program. So we are profoundly grateful for everyone that has put up with our telephone calls and has reached out. Without this, we would never be where we are. Thank you.
Thank you, Susan. Last but not least, Chad Glatzer, the Senior Director of Clinical Research at Tento Therapeutics, is going to tell us about their program.
Thank you, Amel. As others have said, I want to thank the UMDF. This is a great opportunity for all of us to get together and provide all these exciting updates. We're excited to be a part of it. So as Amel said, I'm Chad Glasser. I'm a Senior Director of Clinical Research at Tento. We are a relatively new company, so I do want to spend one slide telling you a little bit about who we are, and obviously give you some updates on a clinical study that we are currently conducting, and then an interventional study that we look to kick off next year. Next slide. So a little bit about us. We are a clinical stage biotech company focused on, of course, developing medications that address significant unmet needs. And our initial focus is on MELAS, and we do hope to expand to other genetic mitochondrial diseases. Although we are a relatively new company, we are working on a molecule that has been studied and hopefully known in the mitochondrial disease community for a few years now. It's currently called zigosiguat, but it was previously called CY6463 because it was developed by Kreon Therapeutics. And a study was conducted that showed rapid pharmacological effects and a really nice safety and tolerability profile. And that was in a 29-day open label study. And that study and the results from that study really was the impetus for spinning out Tento Therapeutics and having this be our lead program and our lead focus. Just a little bit of background about the molecule. It is oral. It's administered once daily. It is a soluble guanylate cyclase stimulator. And you might be familiar with this mechanism because there are a couple other SGC stimulators that are approved. But the difference between those molecules and zigosiguat is that those are restricted to the periphery. They don't get into the brain, and zigosiguat does. It's brain penetrant, so we call it first in class. So it is able to modulate the nitric oxide pathway. Going back to what Dr. Scaglio was saying earlier in his presentation, we hope that this will help address some of the dysregulation that we believe is there in patients with MELAS. We're a relatively small team. We have 13 internal employees. We've been fortunate enough to secure a nice amount of funding to kick off the company and get us through the next key milestone, which is readout from this phase 2b study that we're planning for. We're currently headquartered in Cambridge, Massachusetts. And you can see on the right-hand side of the slide, because we are new and because we're relatively small, we're trying to build an external network that's on a global scale. So we've spoken to, I'm sure, many of you listening, lots of physicians and advocacy groups, of course, UMDF. We have a great partnership with them. Other vendors that have rare disease experience, especially Mito experience, to help us conduct some of these studies. Any other research collaborators out there. So we're spending a lot of time right now trying to build this network and do it really, really well. Next slide, please. So where are we right now? As I said, we are really, really committed to moving as quickly as we can and evaluate this molecule in patients with MELAS. And when I say MELAS, I mean classical MELAS, patients that do have strokes or focal seizures or CNS symptoms that are beyond, MIDD or some of the other mixed phenotypes. But before we kick off this phase 2b study, we know that we have a lot of work to do because it is very challenging. Going back to what was just discussed about heterogeneity of the population, finding the right endpoints, making sure you have the right patients, we have a little bit of work to do up front. And so we like to think that patient perspectives are at the forefront of some of these decisions that we're making because the patient voice is really probably one of the best ways to address some of those challenges. And going back to our name, Tento actually means I hear you in Italian. So patient voice is important. So right now we're doing number one here, which is understand the burden of disease. So we're conducting a patient interview study to identify signs and symptoms, health-related quality of life impacts that these patients are experiencing. And understand, what are the concepts of interest that these patients care about. And we're debriefing a couple scales that are focused on fatigue and cognitive impairment. And again, we're enrolling what we believe is a true population of classic MELAS patients. We'll take all of that data, develop the outcome measures, and finalize our endpoint model. And that will be used to, finalize the design of our phase 2b study. This study, we hope, will be patient-friendly. Similar to what Magnus was saying earlier, we're trying to incorporate remote visits and make this really easy for patients to do these assessments at home. We realize that's very important for trial success. We're trying to establish treatment effects. We're trying to determine what our phase 3 dose will be in a subsequent trial. And of course, all the challenges of endpoint development, we really need the data from the study to do the psychometric work, to validate the measures and incorporate them and get regulatory buy-in. So right now, our focus is on enrolling the patient interview study. It is active. We're looking for patients with MILAS that are 16 years of age and older, that are English speaking. So we've had a really nice influx of patients so far that we're excited about, but we still have a little bit of ways to go. So if you're listening, if you know of patients or you're seeing patients that could be interested, I'll refer you to our website or to UMDF. It's advertised there. And then kind of going towards our phase 2b study, like I said, we hope to kick that off next year. And we're in the process of building those relationships that I talked about on the first slide, figuring out where our sites might be, who are the clinicians and investigators that we want to partner with on that trial. So again, I know we've spoken with many of you and we want to do even more of that. So we'll be doing feasibility work and site selection relatively soon. So be on the lookout for those updates. Last slide is just my contact information. Again, cglasser@tentotx.com if you want to reach out to us and talk to us more about our program. And then our website, tentotx.com, has a little bit more information, again, about the molecule, our company, and then the interview study, which is actively enrolling.
Well, thank you so much, Chad. This concludes our last Bench to Bedside series for 2023. I would like to extend my appreciation to all the speakers today, especially for staying on time. And I would like to remind you that our next Bench to Bedside will be next year on January 8th at 12 p.m. Eastern Time, and will be about neuroradiological features of mitochondrial disease moderated by our own Amy Goldstein. And with that, I would like to wish you all happy holidays, stay healthy, and thank you so very much to all the patients and caregivers who keep enrolling in these trials. We need you to be more engaged, and we need more companies to be interested in mitochondrial disease so we can get to that cure. Thank you, everyone, and happy rest of your day.